Hello guys, this is chapter 20 of the Immortal Life of Henrietta Lacks by Rebecca Skloot, The Gila Bomb. In September of 1966, a geneticist named Stanley Gartler walked up the podium at a hotel in Bedford, Pennsylvania. There, in front of George Guy and all the other giants of cell culture, Gartler announced that he had found a technical problem in their field. He was at the second Decennial Review Conference on Cell Tissue and Organ Culture with more than 700 other scientists. They had come from biotech companies and academia. They had traveled from New York, England, the Netherlands, Alaska, Japan, and everywhere between to discuss the future of cell culture. The room buzzed with excitement as everyone talked about cell cloning and hybrids, mapping human genes, and using cultures to cure cancer. Few there had heard of Stanley Gartler, but that was about to change. Gartler leaned into the microphone and told the audience that, in the process of looking for new genetic markers for his research, he had found that 18 of the most commonly used cell cultures had one thing in common. They all contained a rare genetic marker called glucose-6-phosphatidehydrogenase, a G6PD-A, which was present almost exclusively in Black Americans. And even among them, it was fairly rare. I have not been able to ascertain the supposed racial origin of all 18 lines, Gartler told the audience. It is known, however, that at least some of these are from Caucasians, and at least one, Gila, is from a Negro. He knew this because a few months earlier he had written George Guy, I am interested in the racial origin of the person who your Gila cell line was initiated. I have checked a number of the early papers describing the development of the Gila cell line, but have not been able to find any information pertaining to the race of the donor. When Guy responded that Gila cells had come from a colored woman, Gartler knew he had found the source of the problem. It seems to me the simplest explanation, he told the audience, is that they are all Gila cell contaminants. Scientists knew they had to keep their cultures free from bacterial and viral contamination, and they knew it was possible for cells to contaminate one another if they got mixed up in culture. But when it came to Gila, they had no idea what they were up against. It turned out Henrietta's cells could float through the air on dust particles. They could travel from one culture to the next on unwashed hands or used pipettes. They could ride from lab to lab on researchers' coats and shoes to ventilation systems. And they were strong. If just one Gila cell landed on a culture dish, it took over, consuming all the media and filling all the space. Gartler's findings did not go over well. In the 15 years since George Guy had first grown Gila, the number of published articles involving cell culture had more than tripled each year. Scientists had spent millions of dollars conducting research on those cells to study the behavior of tissue type, comparing it one to another, testing the unique responses of different cell types to specific drugs, chemicals, or environments. If all those cells were in fact Gila, it would mean that millions of dollars had been wasted, and researchers who'd found that various cells behave differently in culture could have some explaining to do. Years later, Robert Stevenson, who became president of the American Type Culture Collection, described Gartler's talk to me this way. He showed up at that meeting with no background or anything else in cell culture and proceeded to drop a turd in the punch bowl. Stevenson and other members of the Cell Culture Collection Committee sat stunned in the audience as Gartler pointed to a chart on the wall listing the 18 cell lines that had been contaminated by Gila, along with the names of the people or places he had gotten them from. At least 6 of the contaminated lines came from ATCC. Gila had penetrated Fort Knox. At that point, the ATCC's collection had grown to dozens of different types of cell, all guaranteed to be free from viral and bacterial contamination, and tested to ensure that they hadn't been contaminated with cells from another species. But there was no test to see if one human cell had contaminated another, and To the naked eye, most cells growing in culture look the same. Now, Gardner was essentially telling the audience that all those years researchers thought they were creating a library of human tissues, they'd probably just been growing and regrowing HeLa. He pointed out that a few years earlier, when scientists started taking protective measures against cross-species contamination, such as working under sterile hoods, it had suddenly become harder to grow new cell lines. And in fact, very few new human cell lines have been reported since. Not only that, he said, but there had been no new examples of so-called spontaneous transformed human cell cultures since. Everyone in the audience knew what that meant. On top of saying they would possibly wasted more than a decade and millions of research dollars, Gardner was also suggesting that spontaneous transformation, one of the most celebrated prospects for finding a cure for cancer, might not exist. Normal cells didn't spontaneously become cancers, he said. They were simply taken over by HeLa. Gardner concluded his talk by saying, where the investigator has assumed a specific tissue of origin of the cell line, liver, or bone marrow, the work is open to serious question and, in my opinion, would be best discarded. The room sat silent, dumbfounded, until T.C.H. Sue, the chair of Gardner's conference session, spoke. Sue was a University of Texas geneticist whose earlier work with HeLa and other cells had made it possible to discover the correct number of human chromosomes. A few years ago, I voiced some suspicion about cell line contamination, Sue said. I'm happy about the paper by Dr. Gardner, and I'm sure he has made many people unhappy. He was right, and those people quickly began asking questions. How long did you keep them in your laboratory? One scientist asked, suggesting that Gardner had contaminated the cells himself after they arrived in his lab. They were analyzed before being grown in my laboratory, Gardner responded. They didn't send them to you frozen? The scientist asked, knowing that contamination could have occurred while they thawed. Gardner said that didn't matter. The cells didn't have to be thawed to be tested. Another scientist wanted to know if the similarity Gardner was seeing between cell lines was just the effect of spontaneous transformation making all cells act the same. Eventually, Robert Stevenson of the Cell Culture Collection Committee spoke up, saying, It looks like more detective work is needed to see whether we're going to have to start all over again to isolate some new human cell lines. Sue stepped in and said, I would like to give particular priority to those who initiated the cell lines, whom Dr. Gardner has attacked. If there's any defense, we would like to hear it. Harvard's Robert Chang, whose widely used Chang liver cell line was listed as a HeLa contaminant on Gardner's chart, glared from his seat. Chang had used those cells to discover enzymes and genes specific to liver cells. If Gardner was right, and the cells were actually from Henrietta's cervix, Chang's liver research using them was worthless. Leonard Hayflick had an especially personal connection with his cell line, Wish, which Gardner had listed as contaminated. He had grown it using cells from the amniotic sac in which his unborn daughter once floated. He asked Gardner whether it was possible to find G6PDA in samples from white people. Caucasian subjects with G6PDA have not been reported, Gardner told him. Later that day, in a talk chaired by George Guy, Hayflick delivered a paper on the facts and theories of a spontaneous transformation of cells in culture. Before beginning his talk, Hayflick stood at the podium and announced that since Wish cells supposedly tested positive for a genetic marker found only in black people, he'd called his wife during the break to ask if he was, in fact, his daughter's father. She assured me that my worst fears were unfounded, Hayflick said. The room erupted in laughter, and no one said anything else publicly about Gardner's findings. But a few people took Gardner seriously. Before leaving the conference, Stevenson met several of the top cell culturists for lunch. He told them to go back to their labs after the conference and start testing cells for G6PD, a genetic marker, to see how widespread this problem might be. Many of their cell lines tested positive, including the skin cells George Hyatt had transplanted into a soldier's arm years earlier. Since Hyatt had no HeLa cells in his lab at the time, the cells in his experiment must have been contaminated before they arrived. And though few realized it, the same thing was happening in laboratories around the world. Still, many scientists refused to believe HeLa contamination was real. After the conference, where Gartler dropped that what became known as the HeLa bomb, most researchers kept right on working with the cells he'd said were contaminated. But Stevenson and a few other scientists realized the potential scope of the HeLa contamination problem, so they began working to develop genetic tests that could specifically identify HeLa cells in culture instead of just testing for the presence of G6PD. And those genetic tests would eventually lead them to Henrietta's family. This is the end of chapter 20.